Hi, my name is Areen. I'm the manager of Professor Uri Hirschberg System Immunology Lab. And today I will start by introducing our immuneDB system and how incantation is integrated in our pipeline. And then my colleague Muhammad will continue and give an example from his work. So this is the view from the System Immunology Lab at the Haifa University, where we study diversity and specificity of the human immune system, as well as its function in health and disease. And we do so by exploring B and T cell repertoires. And as you probably all know that the field of handling immune data is very complex and challenging. And we find that having a tool like immuneDB can be very useful and can help us address this complexity and ask questions about all the level of this system. So when we look at the biological system, we look at it in several levels. We start by looking at the subject data level, from where we take samples that can be from different tissues at different time points. And from the samples, we actually look at bulk sequencing data that we take, and we align and compare to a germline in order to be able to look at mutation and study mutations. And from there, we build clones, we define clones, and we build lineages. And what is important here is that we keep a tight link between all these levels and their metadata. And this is something that gives us the possibility to investigate the repertoire and the diversity. So all of that is practically translated in our immuneDB pipeline. So immuneDB is a database-backed system that analyzes and stores large amount of high-throughput sequencing data from B and T cells. And the database is built in a way that it have tables that stores the data from the levels that I mentioned before. And these tables are linked together in a way that gives us maximum information. So there is a relationship between all the tables, as you can see from the schematic view here. And in addition to storing the data, the database have also tables that contains information from analysis that is done throughout the pipeline, such as the calculated statistics and also a definition of clones. Here is a schematic view of the immuneDB pipeline, where we also integrate incantation in it. So what I want to emphasize here first that is the variety of possibilities to import data into immuneDB. So one can choose to start from a raw FASTA or FASTQ files, or even pre-annotated sequences from an array of formats like change all, or even import air format, where we now have the IGGlass tool integrated in the pipeline. In addition to the variety of importing data and interfacing immuneDB, there are several ways on how one can choose to analyze the data, whether it's in the methods of the alignment tools or in the methods, in the different methods that one can choose in the clonal assignment. Either way, there is a downstream analysis that can be made, such as selection pressure, where we here integrate incantation to baseline as an incantation tool. And there is a possibility to construct lineages, and also there is a possibility to easily interact with the database by importing data in different formats. And also we can interact by having the ability to make queries to a REST API, where immuneDB is now part of the iReceptor gateway. In addition to all that, immuneDB has a front end, a web interface, a very interactive visual way to view the data and to look at the basic analysis that are made through the pipeline. And here I would like to show an example of how the web interface, how the front end look like. So here's an example of the of a data set of the COVID vaccine data, where we can see here all the samples separated by each subject, and we can choose to group these samples by any metadata information that we have. We can choose to make a quick analysis. We can choose as many samples as we want and make a quick analysis to view this information. And here we can see information such as VG usage, CDR links, copy number, graphically, and in a very informative way. And also we can choose to make a custom analysis, where we can look at the clones in different formats. We can choose to look at it in different formats, whether it's immuneDB, VDJ. Also choose to import the selection calculations, sequences, and other things. And also we have a possibility to look at clones, choose specific clone and look at it, look at all the information that are linked to this clone, all the mutations divided by each region, CDR1, CDR3. You can visually look at the mutation, which is really nice. In addition to the mutation, we can have a look at the tree, at the lineage, in also an interactive way, and choose to look at each node and see which sequences are contained in each node. For example, we can see here that there is an interesting node that have sequences from two types of cell subsets overlapping. So you can see that this might be a very useful information. And this is all. So I will finish by and summarize things that I said, and introduce why it's very useful and why we recommend using immuneDB. So first, it allows us to focus on our experiment and easily share data and look at it visually through the interface, through the website. And also immuneDB knows how to save clonal annotations, unlike most available databases that only save the raw sequences and rearrangement without any link to the cloned, which is something that give us the ability to investigate the diversity and the changes in the diversity due to the association with metadata, and actually be able to make complex queries on the clones, as well as the special architecture of the database, of the immune database, that makes it very easy to query, for anyone to query and study the immune repertoire, its characteristic, and also be able to make any changes along the way without the need to create the annotations from the beginning. Now, Mohammed will continue.
Hi, everyone. The example I want to talk about is a collaboration study with Shlomchik lab of memory subsets CD45, the B-cell differentiation. What we would like to know if there is an order of formation of these memory subsets. We sampled 3 individuals from the spleen tissue. All cells are memory cells and are differentiated into CD45 subsets. As I said, we want to see if we can identify an evolution in the formation of the memory subsets. How do we do that? We take B cells, mutants, and we order them in lineages. And then we ask, do we often see that one subset is formed after the other one? For example, if we think the CD45 positive subset is formed because some kind of selection events after its parent, the CD45 negative, then we expect to find the negative one more often before the positive one. To do so, Immcantation offers the test edges tool. The test edges, it performs a permutation test on a set of lineage trees to determine the significance of an annotation's association with parent-child relationships. In our case, the annotation would be the subset type, the CD45. In general, a permutation test involves randomizing the data in some way and comparing the observed test statistic to the distribution of test statistics under the null hypothesis. The null hypothesis in this context might be that there is no association between the CD45 positive and the CD45 negative. And this is done to assess whether the observed result is statistically significant. But in order to use the test edges, you need 3 lineages, clones, which is iGraph object containing an annotated lineage tree. So to do this analysis based on Immcantation tools, first of all, you need to start with your raw data, which would be the error or the change O data. Then you do your clonal assignment and formatting by using Immcantation tools. Then you need to get your trees, and to get that by Immcantation, you could use one of their packages, the IG female, the Philip, or using our packages. Then you can start using your test edges test. So, our pathway, you can either start just by downloading the immuneDB change O clones, and then use Immcantation get trees methods, but it's a little bit slow. So you could use the second option by just converting our immuneDB hierarchical trees to the appropriate format, the iGraph, and then you give it as input for the test edges. As Areen showed you, each clone in immuneDB is stored in a hierarchical JSON tree chair. Here is one line from the immuneDB database for this clone. with the ID 129694. We converted the stored JSON hierarchical clones to iGraphs with the needed format for test stages, with making sure to save the hierarchical order. So here is a part of the results for the 3 subjects. Unfortunately, no ordering was detectable, as we see, in the progression of spleen memory cells. The p-value of the results of the switchings between positive and negative are not significant. It's not highlighted with the green, which means we didn't identify some kind of relationship or selection pattern of subset type for positive to negative or vice versa. To conclude, we already presented immuneDB, which is a system storing and analyzing high throughput immune receptor sequencing data. We suggested a method to use test edges without using Immcantation pipelines for clonal assignment. And we encourage you to use immuneDB and recommend Immcantation, a team, as well as us to adjust more cross-talking pipelines between Immcantation and immuneDB. And thank you for the listening.